Okay, so the circulatory system. It's actually composed of two parts. Of course, the cardiovascular system, which is carrying the blood, contains the heart, the arteries, the capillaries, the veins, and then the lymphatic vascular system, which collects and returns excess interstitial fluid to the blood. So typically, we lose about 20 liters of the liquid component of our blood into the interstitial spaces. And then about 17 liters is reabsorbed by the small blood vessels. But then the lymphatic system picks up that last 3 liters, drains it through a series of tubes, and dumps it back into the blood circulation. So, of course, it also has important immune functions, but we're not really going to talk about those until November. Okay, so throughout the cardiovascular system, or the blood system, there are 3 layers. In the heart, we have the epicardium, which is the outermost layer, the myocardium, which is the muscular layer, and its associated endomysium and perimysium, and the endocardium, which is the innermost layer that lines the inside of the atria and the ventricles. In all but the very smallest blood vessels, there are also 3 layers: the tunica adventitia, the outermost layer, the tunica media, the middle layer, and the tunica intima, which is the innermost layer. So let's start with the heart. So in this diagram, if you had this drawing is like a little cutout of the heart. So if we look up here, so this would be the lumen of the heart, and then the little white line would be the endocardium lining the lumen of the heart. So of course, quite thin, you can see. The thickest layer, the myocardium, the muscle, and then the epicardium here on the outside. So that's a little thicker layer. It contains quite a number of different tissues. And the epicardium is also called the visceral pericardium. So, you know, the heart is covered by sort of a fibrous layer called the pericardium. And then there's like a little cavity in between this outer fibrous pericardium and this inner epicardium that's filled with some serous fluid that prevents friction. So this outer layer is called the fibrous pericardium. This inner layer is called the visceral pericardium or the epicardium. So the epicardium and the visceral layer of the pericardium are the same thing. So starting with the endocardium, we're going to start on the inside and work out. So down here you've got the myocardium. I think you probably are all recognizing that these are cardiomyocytes. And then the endocardium is composed. Is that a laugh? Okay, look. I see some intercalated discs here. We'll just stop for a moment and talk about it. Intercalated discs, look, centrally placed nuclei. These aren't really cut longitudinally or in cross sections, so they're a little hard to see, but you can see that they're filled with all these myofilaments, and I guess we can't really even see the striations here. Yeah. These little pink lines here. Okay, those are intercalated discs. Okay, so the myocardium, and of course in between all the cells we have this fine sparse connective tissue called the endomysium. Yeah? Okay, so the endocardium, it's composed of a simple squamous epithelium called an endothelium. So that simple squamous endothelium, throughout the cardiovascular system, those lining cells are going to be called endothelium. Occasionally they look a little cuboidal. They're underlined by sort of a layer of loose, irregular connective tissue, sort of sparse fibers. And then underlying that, we see a subendocardial layer. Sometimes it's very thick, sometimes it's very thin. Sometimes you'll hardly even see it there. But if you see it, it is loose connective tissue. It might have some blood vessels in it. And in the ventricles, it will have the Purkinje fibers. Do you remember we talked about the Purkinje fibers that help conduct the signal to beat or the signal to contract around the ventricles? Look, and so you remember that they are modified cardiac fibers, and look, they even have intercalated discs. Here they are. Okay? So, yeah. So these are the Purkinje fibers. So you see how they look a lot like cardiomyocytes, right? They're just bigger. Their myofilaments are just, they look sort of wispy, and they're not arranged right next to each other. And these cells are much bigger usually than the cardiomyocytes. So in the subcardium, we also see some connective tissue. Sometimes it's dense, irregular. Sometimes it's loose, irregular. We often see a little bit of smooth muscle there. So here's another example. In this example of the endocardium, look, we can see the little endothelium cells. We see their little nuclei sitting right here on the surface. Okay? And so here you can see the Purkinje cells pretty clearly. So they're quite large cells. So look how big they are compared to, I mean, you can't even really see whole cells here, right? But they're really big, centrally placed nuclei. Look, some intercalated discs right here, right here. Okay, one more view. Here's the myocardium. Here's the endocardium. My little arrows don't look great there, but so here's the endothelium, these little cells on top. Here we've got this loose connective tissue lying underneath and some subendocardium down here. So if you were to get an identification question on an exam, you would not be seeing an image like this. You would be seeing more an image like this, you know, and you would need to know, oh, look, the epicardium's here, the myocardium's here, and there's going to be an endocardium here. Okay, so that's what we have, an epicardium, a myocardium, and here's the endocardium. Here's a little blow-up of that, just one more view. Endothelium, connective tissue, Purkinje fibers, a few smooth muscle cells sometimes. All the images that I've shown you, it's quite thick looking, but sometimes it looks quite thin. Sometimes you would hardly see more than this little layer here. Okay, and of course you won't see Purkinje fibers in the atria. Okay, so here we've got some myocardium. We've got some cardiac muscle cells with centrally placed nuclei. You aren't able to see any striations here, but you know that they're there. And you know that they're connected by intercalated discs. Uh-huh. Yeah, it sort of depends on where you are. It might look sort of dense, it might look sort of loose. And as you remember, that's a little bit of a judgment call, right? I mean, you're just going to look at it and see what you think. Unless, I'm sorry, we're going to change that. You know, in certain tissues, you know, like you're going to look at it and we're always going to say this is loose irregular. So say right underneath the very surface of the epidermis, we're always going to say that. And then deeper, we're always going to say it's dense. Okay, the epicardium. It has a pretty characteristic appearance. So here's the myocardium again. And here's the epicardium right here. You can see it's sort of a more substantial layer. It has a simple squamous epithelium. It's called a mesothelium when it's here on the outside of an organ. Okay. This is actually the pericardial cavity. And then this is the parietal, or the outside portion of the pericardium. So underneath this mesothelium, we've got some irregular connective tissue. It's very typical to see nerves coursing through that area. So this is a nerve. You see lots of fat in this area, so you can see some adipose tissue here and here, here. You'll always see blood vessels there, smaller blood vessels. Here you can see a blood vessel leaving the epicardium and moving into the myocardium. So thick. So to me, it always has a little looser appearance. It always has a lot of fat. And you're always going to see nerves there and blood vessels. So the valves of the heart are made of dense irregular connective tissue, probably not surprising to you. They are covered with the endothelium. So here we would have epicardium, myocardium. There'll be endocardium lining here, and then it's going to come around and wrap around this valve here. So here's a micrograph of that. So you can see on a thin layer of endothelium, see those little purple nuclei sort of poking up. So that's the endothelium. You can see it on the other side too. And there's some connective tissue underneath, but it's just not really evident that it's different from this really fibrous connective tissue, lots of collagenous fibers, big bundles that the valve is composed of. Okay, so just in summary, epicardium, flat tooth cuboidal epithelium, called a mesothelium, that is sitting on fibrous and adipose connective tissue containing nerves and blood vessels. The myocardium, the thick striated cardiac muscles. There'll be some blood vessels, and you'll see the connective tissues that we talked about earlier, the endomysium and the paramysium. And the endocardium, a simple squamous to cuboidal epithelium called an endothelium sitting on a loose connective tissue layer. In the ventricles, it will contain the Purkinje fibers. So the endothelium is actually consistent or continuous with the tunica intima of the large blood vessels leading the heart. Okay, so any questions on heart before we move on? Yeah. I'm not quite sure what you're asking. In here? Uh-huh. Yeah, that's just going to be connective tissue there. Okay. Okay, so the walls of all blood vessels except capillaries contain smooth muscle and some connective tissue, of course, and are lined with simple squamous epithelium called an endothelium. So here's an example of a small blood vessel. The smooth muscle in arteries is always wrapped around it circumferentially. So these are smooth muscle cells here. And so this is cut longitudinally so they look round. Remember, they've got these long oval nuclei, but if you cut them in cross-section, they end up looking round. So that's what these are. These are smooth muscle cells. So of course, here's the lumen with some blood vessels. And then here's the endothelium, so these flattened nuclei. A lot of times in arteries or arterioles, those actually pop up some, but not always. See, they have this sort of very thin, attenuated cytoplasm. I think you can see some right here, and here, some right here. So they're little cells, but they are doing so much. So they have so many functions besides just conveying the blood through it. So they are very active in controlling movement of molecules across that barrier. So lipid-soluble molecules and oxygen can diffuse right across it, but almost anything water-soluble is going to have to be transported. So say glucose, amino acids, they're all going to have to be transported across by active transport or a variety of endo or pinocytosis. And I think you're going to have a lecture either today or tomorrow, later today or tomorrow, that's going to talk about this a lot more. They help control clotting. So typically, they present a non-thrombogenic surface so that obviously clots don't form within the blood vessels. In response to injury, they can release factors that, in fact, encourage clotting. They release a variety of factors that can cause vasodilation or vasocontraction. So they help control blood flow. They help control blood pressure. They play a role in regulating inflammation and immune response, largely by regulating the entrance and exit of cells of immune cells from the blood vessels. So those cells usually exit actually in between endothelial cells by special processes, and we'll talk a lot more about that when we get to the immune system. They secrete various growth factors. They're important in angiogenesis. They modify lipoproteins in the blood to tell the body it's time to remove these. They're getting old. So this is all those things I just told you that the wonderful endothelial cells do in more detail. This is really just for your information and because I thought it might be useful later in your career here. Okay, so let's look more closely at the blood vessels. So, of course, the heart is the pump. The largest arteries that come out of the heart are called elastic arteries. They branch into muscular arteries and finally into arterioles. All of these have these three layers, the tunica intima, the media, and the adventitia. Some authors call this the externa. So a tunica externa is the same thing as a tunica adventitia. The capillaries, which are just one layer of cells, just endothelium, and then back up the venous side from small veins, which we call venules, to medium and large veins. Okay, so except for the capillaries, all the walls here have three layers, the intima, the media, and the adventitia. So that is usually the thinnest layer. So, for example, in the artery here on your left, the pink and the tan are the intima. And the vein here on your right, again, the pink and the tan are the intima. And the first thing you notice is that the lumen of the vein is much larger than the artery, and that's pretty typical, is that the lumen of the veins is much larger. The media, so the intima is usually the smallest layer. The media, in an artery, the media is always the biggest layer, and it's typically much larger and more substantial than you see in a vein. And finally, the outer layer, the adventitia, it's actually much larger in a vein than it is in an artery. And so, of course, capillaries only have endothelium with no other subendothelial layers or tunics. Veins also have valves that arteries do not, to prevent backflow of blood. Okay, so this is actually a micrograph that has been so heavily augmented with crayons that it just looks like a drawing now. But it still shows you the point that I want to make here. So arteries tend to be round, whereas veins tend to be sort of flattened. The artery has a large media, so this dark pink part is the media, and here's the artery. There's a vein, small media, and the adventitia is typically, it looks pretty big here, but it's typically larger in the vein than it is in the artery. So conveniently, arteries and veins tend to run side by side, so you almost always have a comparison right there by ear. So this slide is partly for study, but I just want to go over some of the important differences between elastic arteries, muscular arteries, and arterioles. So these are, of course, the largest. The intima of muscular and elastic arteries looks fairly similar, except that on a muscular artery, there's this really thick layer on the outside of the intima called an internal elastic lamina, and we're going to see that. So this is one of the major things you're going to use to distinguish that. It also has a really thick layer on the outside of the media called an external elastic lamina. The tunica media of elastic arteries is filled with these sheets of elastic tissue. Some call it elastic membranes or lamellae, but they're these sheets of elastin that are quite thick, they're perforated, and they're very prominent in these elastic arteries. Okay, and both of these vessels have, in the adventitia, they will have quite a few blood vessels that run through there, and that's important to remember. Arterioles are really easy to see. They have a small intima and then one or two layers of smooth muscle cell, and so that really makes them easy to pick out, and an ill-defined adventitia. Okay, so let's start with elastic arteries. So elastic arteries have an intima. So the intima goes from about here to here. It's an endothelial layer with some connective tissue and smooth muscle cell underlining it. And then it has an incomplete internal elastic lamina, which is not indicated here very clearly. But now when we get to the media, all these yellow lines, these are the elastic laminae. They are, even though very often you need a special stain to look at elastic fibers, in an elastic artery you don't. They're just so large and so prominent. So in the adventitia, it is largely connective tissue, lots of big collagen bundles, some elastic fibers which are much smaller than these. Typical things you see in connective tissue, maybe some macrophages, but you're also going to see nerves, and you're going to see blood vessels. And the blood vessels have a special name. They're called the vasa vasorum, so they're vessel of the vessels, and they supply nutrients to these large blood vessels, to the adventitia and the media. So elastic arteries are the arteries that are closest to the heart. So I think you can imagine that when the heart beats and this big bolus of blood comes out, the blood pressure would rise. So it expands, and those elastic fibers help it to expand. And then in between beats, it can get smaller, and so that sort of helps equilibrate the blood pressure within this large elastic artery. So that would be the aorta, the pulmonary arteries, the carotids, maybe just a few of the vessels that start branching off the aorta. Okay, so here's an example. So the lumen's up here. So the intima's here on the top. It's quite narrow. And here's the media. And here's the adventitia. So let's look at the media first. Can you guys see there are all these little lines? This is just an H&E. It's not even a special stain. Look at these lines. Can you see these little squiggly lines? Those are these elastic membranes or elastic lamellae. And you can see they're so big that you can see them at an H&E level. They're fenestrated, which means they're holes, but they're like sheets, really. In between these lamellae are mostly smooth muscle cells, lots and lots of smooth muscle cells. In arteries, smooth muscle cells are arranged circumferentially. So here you're largely seeing them cut in cross sections, so their nuclei are looking round. There's a few collagen fibers, some other fine connective tissue elements, but the big components are these elastic membranes and smooth muscle. So I think you can see here in the intima next, there are a few endothelial cell nuclei here at the top. The distinction between the intima and the media is sometimes a little hard to see. There's this first internal elastic membrane, but it's quite indistinct in an elastic artery and difficult to find, and it's also discontinuous. So there's an external elastic lamina that separates the media from the adventitia. I think you can see when you get to the adventitia, there are these big bundles of collagen. It looks quite different. And there are these blood vessels, the vasa vasorum. So we're going to look at this box and this box blown up, higher power. Okay, so here's the intima, here's the media. These are the nuclei of endothelial cells sort of popping up. There's some connective tissue. This is a smooth muscle cell. And then there are a lot of fibers. And again, the distinguishing between the intima and the media is a little of a judgment call. Okay, so now we're in the media. And it's a little harder to see those elastic fibers at this magnification, but like here's a big bundle of this elastic membrane. Got a lot of smooth muscle cells. This one got cut lengthwise, so you can really see its cell body here and its characteristic corkscrew nuclei. Okay, now we're going to look at number 3 here. Okay, so we've got the media. We've got a big smooth muscle cell still here. So I would say that this is the external elastic membrane here. And now we've got lots of big bundles of collagen fibers. We've got these little dark nuclei that belong to fibroblasts that are helping us make this collagen. And here's a blood vessel, a small blood vessel, cut in cross section. Here's the lumen. Here's the nucleus of an endothelial cell. And here's the media that contains these 2 smooth muscle cells. Okay, in this preparation of an elastic artery, you can't even see any cells, but it's done so that we can see the elastic lamina so clearly. So you can really see how extensive they are here in the media. In between, there would be smooth muscle cells and the intima and the adventitia, and you can see a few blood vessels, vasa vasorum down here in the adventitia. Okay, so as the arteries branch and get a little smaller, they lose these elastic lamellae that are in the media and become muscular arteries. So their media is largely filled with smooth muscle. That's what you see here. But they become easier to distinguish, at least for me, because their internal and external elastic lamina become so prominent. So there's the internal elastic lamina, the intima on the inside. Of course, this would be the lumen. You've got smooth muscle here. It's pretty filled up with smooth muscle. There's still a few elastic fibers. Here's one here. There's one here. But they're not these huge sheets of elastic membranes. You can see that the external elastic lamina is also pretty apparent, although not quite as apparent as the internal. So then here's the adventitia filled with irregular connective tissue, It still has a vasa vasorum, big bundles of collagen fibers, lots of fibroblasts. Okay, one more view of a muscular artery. So we've got an artery and a vein running side by side here. So here's the artery, and here's the vein. So we'll come back and oh, all right, that's not an option for me today. What? Okay. Well, next time it comes up, we'll do that. Or we'll see if that's an option that is open. Okay, we'll come back and we'll look at the vein a little later, but we'll just look at the artery right now. So this is just an H&E, no special stain for elastin. But look, even on an H&E, that internal elastic lamina is so prominent. And of course, the intima, it's looking pretty sparse here. On the inside, you can maybe see a few endothelial cell nuclei here. The media, the thickest layer, filled with smooth muscle, a few collagen fibers, and finally the adventitia from here to here, thinner than the media, big bundles of collagen fibers, and there will be blood vessels in here as well. Okay, so the next smaller size is arterioles. So arterioles consist of an endothelium. See here the nuclei sitting on sort of some scanty connective tissue. They have usually a very indistinct internal elastic lamina. This is a surprising micrograph. So this is actually a plastic section. That's why it looks so crisp compared to a paraffin section. And then it's cut longitudinally, so we're cutting this smooth muscle in cross-section. So we see two layers of smooth muscle cells here surrounding that arteriole. So 1 to 2 layers of smooth muscle in the media of an arteriole. And then the adventitia is sort of an indistinct sheet of loose connective tissue. That's out here. So here's a cross-section of an arteriole. So the lumen, of course, the endothelial nuclei, they're sort of popping into the lumen. Again, that's a very typical appearance.